I have been asked to talk about gene therapy and what families should know, and I'm very happy to do so because when I showed some results of the gene therapy in Rome at the Parent Project meeting in February, I described this as being a roller coaster because this has really been a lot of emotional involvement, very high expectations, a lot of disappointment, and then now we are again in the phase up when we have some results, but. This has been a very confusing field, a lot of rumors, a lot of things happening. And so I have tried to make a sort of short history of gene therapy and where we stand now. I have some disclosures that I forgot to put here, but I have some disclosures because I have been involved in the Pfizer study, in the Sarepta study. We are about to start with the study from Solid, also from Genesis. So I have been involved in most of the gene therapy studies, and I have been in advisory boards with all these companies. So what I try to summarize today is what we know for in Duchenne and in gene therapy in Duchenne. To talk about a bit safety, there is a lot of concern, and also about efficacy. There is a lot of confusion about this efficacy, but also to bring together some lessons we have learned from our experience with the trials, and now with real world evidence of gene therapy. Just in general, to say that when we talk about gene therapy in neuromuscular disorders and specifically in Duchenne, we talk of a gene which is in a transgene which is inside a viral vector, an AAV viral vector, adenovirus associated, that is important to bring the transgene inside the muscle cell, to enter the nucleus and to release the informations which are important to producing the missing protein. Now, the issue is normally we have one factor which has the whole gene inside, the whole, and this is, however, not possible for dystrophin because the dystrophin gene is the largest gene we have in our body and does not fit into the size of the vector. So the dystrophin gene is too big, and so this has to be shortened. And what you see in the lower part of this slide is an example of how the whole gene looks like. It's a very big gene with all these exons divided into domains and so on. And the second is the dystrophin gene in a Baker patient who was older than 55, if I remember correctly, and he was still able to walk. So you can see that there is a big missing part. There are a lot of exons which were not there. But even if all these exons were missing, this person was still able to walk. And this was an important lesson because we started thinking, and there are many studies on Bakers, that there are parts of the gene which appear to be less relevant than others. So based on all the literature on the Bakers and so on, several companies have produced different gene therapies where they have cut the gene and made a mini or micro gene which is producing a mini or micro dystrophin. And as you can see from this slide, the cuts that were made were not identical, and also the vectors they were using, they were not identical. So we have a number of choices, starting from the first one, from Sarepta, Roche, Geneton, Regenex Bio, Solid, and the Pfizer one, which are all different from each other. And the first question is about safety, and what families should know is that safety, as well, you already know, is the most important thing. But I want to stress how important to understand the safety, the monitoring. Well, the first thing we do before we enter a gene therapy is that we go and test if the antibodies against the virus are already there. So the first thing is to assess if antibodies are already there. And because the child can already been exposed to the virus either because the virus exists in nature, so for an environmental exposure, or especially if patients, children, boys have already been receiving other gene therapies or other drugs which have an AAV component. So it's important to remember that patients who have been treated with an AAV gene therapy cannot receive a second one as things are now, but also this is not the only criteria, because sometimes and there are some countries where there is a higher risk of being exposed to the environment. When we had the first trial with Pfizer, I remember that there were countries, I think there were Spain, but also in Italy, we had a quite high percentage of patients who were already positive to the antibodies. And then it's important to remember that it's not only the immunity that we already have that is important to determine a possible response when we do the dosing of the transgene and the vector, but also there is an acquired immunity that can happen at different times. And so it's important that we understand about the preparation and we understand what are the possible safety effects that we are expecting. And there are ways that as clinicians, we have to reduce the possible risk, like checking for infections at the time of the dosing, or checking for possible immunological abnormal responses that may be there already before the dosing that we could explore better. What has been done over the years based on the results of the clinical trials, not only in gene therapy with the drugs that we have been using, but also especially also in SMA and so on, is that we can have a calendar of what are the possible adverse events that can occur and when they may occur, because these events are related to different types of immunity. The first one are more related to the vector, and the second part are more related to the transgene, and they are mediated by a different immunological response. And this is another way of showing that there was a very interesting NMC workshop that was for all the gene therapy neuromuscular disorders, who made a similar graph. And again, as families and clinicians are aware of what we should expect, and this stressed the importance of monitoring. When we perform a gene therapy that is a one-off, we are always concerned that, because this has happened in some cases, especially in SMA, that some families will sign a consent saying that it will be follow monitoring and so on, but because they don't need to come back to receive the drug again, some of them come from other countries or from other areas of the same country, and they don't come regularly to monitoring because the grandmother is sick or the car is broken. And it's very essential the families understand that each single assessment that we do for monitoring is important, that this monitoring must not be missed. So the safety that I think we have some conclusions about safety in gene therapy. The main message is a very important issue, should never be underestimated. There is some preliminary work done before dosing, that the first days or weeks are extremely important for the immune response to the vector, and we give an extra dose of steroids to make sure that we reduce the risk for the first month. And after a few weeks, there are possible responses to the transgene. And again, monitoring is very essential. What we have seen for safety is that the drug is generally well tolerated. Most of these signals that I have shown you, the liver, the platelets, and so on, can be generally well managed. There have been a few cases where these adverse events has been much more serious, and you are all aware that some children died. In the past, there has been, before we recognize that some mutations were a higher risk, there has been a few cases across all the different programs. Now we know this a bit better, but safety still should not be underestimated, even if we have a better control of the safety. I will move now to efficacy. And as I said, there are a lot of clinical trials which are running. The Pfizer study was unfortunately completed with no evidence of efficacy over 12 months, and we will go back to duration later. And other studies are starting or are have fully recruited, but are not fully completed. The only study which has final results are the one with delandestrogen moxeparvovec, the drug developed by Sarepta and now together with Roche. So the structure of delandestrogen is similar to the other ones we have seen. There is a transgene. There are other parts like the promoter, which are important to focus the activity and to on targets and to determine the expression levels and the specificity, and there is the vector. And this was the EMBARK study, which was the main study after a smaller study. The pivotal study was the EMBARK studies with 125 children who were enrolled. They were 4 to 7, less than 8, and this was divided in treated and placebo. And after a year, there was a shift so that the children on placebo could receive also the gene therapy. When the study was results, when I was talking about a roller coaster, this was partly disappointing. You know, we were all expecting more results, better results, and what was disappointing was that not only you know the results were not as much as we wanted to see, but also that the measure that was chosen as the primary measures, the North Star, did not show significance. And this was upsetting because if we had chosen the time to rise from the floor or the 10-meter, for example, or other measures like the stride velocity of the 4 stairs, they would have all reached significance. So again, sometimes saying that the child is successful or not also depends on the measure, on the design of the study. In this study, as I said, if there are 4 measures that if they had been chosen as primary measures, then today we would be saying that the trial was successful. So this is a first lesson, and we have learned a lot on which measures are important for different individuals at different ages and so on. And I always get upset when I hear even my colleagues who are experts say, the North Star is not working, or this one is not working, because as we will see later, if we consider other age groups or longer duration, then this drug becomes your best friend. So it's very important not to take conclusions based on one study, which was on a specific population, but things should be looked more globally, and we now know much better than the time when these studies were designed how each of these measures can contribute in different age groups. And another important thing, so overall, all this information showed that there was always a positive signals. This line means the line in the middle is 0. This means that everything which is on the right means that the drug was working better than the placebo. And if the responses had been on the left, meant that the placebo was better than the drug. So you can see that irrespective of whether the measures were reaching significance or not, there was always an improvement in 12 months. And this improvement was significant for a number of measures. And it was very nice that when we use something objective like muscle MRI that can measure in an objective way and quantify the fat fraction, there was an obvious difference between the treated children and the placebo ones, and the amount of fat fraction which was much more in the placebo compared to the treated ones. So these results, which are, again, they do not depend on collaboration of the child, they're objective, they're also trending in the same direction of the clinical measures that we saw. And of course, one wants to look at the expression of microdystrophin, because this is the proof of concept, and this is the measure that we use in the US for approving the drug. You can see that there was an improvement in microdystrophin on 3 different measures. And also what is nice, these are the results of the second year, that if you compare the amount between week 12 and week 64, in all the measures, there was always an improvement, which we can argue whether this was relatively small or more, but as a proof of concept, it's important to see that there is also the same trend observed both on MRI, on biopsy, and in the clinical measures. And when I was talking about the need for probably having a better duration, these are the results of the same scale which was blamed at 1 year, the North Star at 2 years. When you look at the results at 2 years, the difference with the placebo is much more obvious and reach a very strong statistical significance. And the same applies to the timed items like the time to rise from the floor and the 10 meters. So hopefully duration will give us better results. And these are the results that we have to interpret with caution, because there were only 4 boys in this in one of the first study that was using adeladistrongen, and you can see that when these data were compared to an external cohort, there was an improvement followed by a stability that was not observed in the external controls. Now, these results have to be interpreted with caution, and when the results are small, I always like to look at the individual results. So in this graph, the purple line is the child, each of them is one of the 4 children, is the results in the treated child, and the bar below is what is expected in the natural history with increasing time. And you can see that in practically all, a bit less in the 4th one, there is some variability, but there is really more stability compared to what the natural history would have said for matched controls for the individual children. So these results seems to suggest that the long-term results are much more interesting, because first we can look at important milestones like loss of ambulation or hand to mouth for ventilation, and but also because we know that with this mechanism of action, dystrophin restoration, we may need more time. And these are examples from other studies. The first 2 columns are about the eteplirsen, which was the first antisense to be approved in the US. And if you look at the results of 12 children who were treated in the first study compared to age and properly matched controls, external controls who were untreated, you can see that in the first year there was hardly any difference between treated and external controls. In the second year, this became more obvious, and with increasing time, this difference became more and more obvious. And if you look at the first graph, we show how many patients lost ambulation in the controls. In the treated, you can see that in the treated there were 2 boys, actually twins, who lost ambulation immediately soon after being treated, so that they were not on treatment very long, while in the external controls there was an increasing number of patients who lost ambulation. Similar results have also been observed using other antisense oligonucleotides like golodirsen, and this applied to motor function and also to respiratory function. And I want to show you also the results from Translarna, where again, when we had different trials in the short term, in the first year, there was again a trend on all the measures, always trending in favor of the drug, but this did not reach statistical significance. But when you look at the results on the long-term registry, you can see that there was a delay in the loss of ambulation of over 3 years compared to a population of controls matched with propensity matching. So there are a number of other challenges. So duration is one, but there are also some items related to age-related scales. We now know that the North Star starts the decline later compared to time to rise from the floor. So the North Star age 4 is not the ideal measure for the very young ones. We know that there is an effect of development on the young boys. Some children start with a delay, and they will acquire the ability to do a number of things like going up, downstairs, because of the brain dystrophin involvement. And also there are a number of other questions. We do not know whether producing mini dystrophins means the same thing of having the same amount of full dystrophin. There are issues about is this going to be persistent over time. We have some results at 5 years, but they're also on 4 boys. So there are still a number of questions that are not fully addressed for gene therapy. And what are the conclusions? That I think we are now in our roller coaster, we are now going up. The long-term results are interesting. The 2 years data are much better than the first year data. We have a much better understanding of the safety profile, but I think this optimist has to be cautious because we don't have results. And I think because this drug is available in some countries like the US, but also in other countries, I know they are using in Russia and in other countries, we really need the clinicians to collect this data, because only from the real world long-term data we will have information on durability, we will have information on efficacy, and we will have information also on safety, because as demonstrated by a recent case, not only we see important signals of safety during the trial, and we better see them when the drug is given in much larger numbers in the real world. So I think and I'm happy to take questions. So we are still in this roller coaster. I hope I set the record straight on some information, because I hear there are a lot of rumors on both on safety and efficacy, which are very superficial and very often fake. So I'm very happy to take questions on these presentations, or more generally in gene therapy. Thank you. I will stop my presentation here.